take a look at some other characteristics of enzymes. So one important characteristic of enzyme needed to do its jobs is the ability to differentiate between similar substrates. So remember that enzymatic reactions happen in the cell, and so there are a lot of different molecules in the cell all at the same time. And so the enzyme needs to be able to pick the correct substrate in order to catalyze the correct reaction. It needs to be able to differentiate that very carefully. So for instance, this is substrate specificity is what this is known as. And so for instance, if we look at this enzyme called TXT, it can take L-tryptophan, which is just your canonical amino acid of tryptophan, and selectively nitrate this. So it adds an NO2 group to the molecule, okay, as the product. So the substrate is tryptophan, the product is this 4-nitrotryptophan. But there are similarly shaped molecules, right, within the cell. So, for instance, phenylalanine, which is also an aromatic amino acid, L-tyrosine. Neither of these are taken up by this enzyme. This enzyme cannot use either of these and perform the reaction on these. So it is very well tuned. The substrate binding pocket is very well tuned towards specifically tryptophan. In addition, this goes to stereochemistry. So the enantiomer of L-tryptophan is D-tryptophan, and the enzyme also can't use this either as a substrate. So again, the enzyme are able to differentiate between similarly structured substrates. Okay. So another example of what enzymes can do is that they can make a specific product. Okay, so in particular, let's stick with this TXT enzyme. This nitro group could end up in a lot of different positions. So this is it could end up in this position or in other positions on the ring. But it specifically, and this is a case of regiospecificity, adds the nitro group to this position on the tryptophan. All right, it doesn't end up in other parts of the ring, or it doesn't add 2 nitro groups instead of 1. It all ends up in one position, and so that's an example of regiospecificity. So the region in which the chemistry occurs is consistent for the enzyme. Finally, enzymes can control redox. So if you remember in organic chemistry, something is very hard to control redox, such as oxidation of carbons or alcohols, for instance, to carboxylic acids or ketones, right? If you remember, or aldehydes for that matter, right? So, but enzymes are really good at this. So an enzyme can take an amine group on this substrate here and convert it selectively to an NO instead of, so this is the nitroso group, instead of under-oxidizing to a hydroxylene group or over-oxidizing to a nitro group. So this is another example of the specificity that enzymes can do. So enzymes can summarize, enzymes can select a specific substrate amongst many choices within the cell. They can form selectively or specifically form regioisomers, and then they can also control the redox or the specific reaction that is going to occur if there are multiple choices or multiple ways this reaction can happen.